In the previous video, we talked about how things might move across the membrane because very few things can actually just move right across the membrane and that they use membrane transporters. But what dictates when something might move across that membrane? So it's very easy to think that when the body is at homeostasis, everything is at equilibrium, right? So if we draw this cell here, it might be easy to say, well, if I tell you that there is sodium inside of the cell, there's sodium outside of the cell, you would be inclined to say that the amount of sodium outside of the cell would dictate or be the same as sodium inside of the cell, because you would say that it's at equilibrium. But one of the things that we have to remember is that homeostasis does not equal equilibrium. Because those cells are selectively permeable, you can end up with situations where there's a high amount of sodium on the outside of the cell and low amounts of sodium on the inside of the cell. And that's how a lot of the cells actually look. And the opposite can be said for potassium, where there's a high amount of potassium inside of the cell and a low amount of potassium outside of the cell. So what's going to drive this sort of movement? And so one of the first things that we talk about when we talk about movement of stuff across a membrane is we tend to talk about diffusion. Okay? And diffusion is movement from high to low, is what we'll say, right? But what does that mean to go from high to low? You've always talked about, well, it's the concentration. Always move from an area of high concentration to an area of low concentration. And that's not always true. When we talk about high to low, we talk about energy, right? They'll move from an area of high potential energy to an area of low potential energy, right? So this is basically a movement from high energy to low energy, and this can be it's a passive process. And what does that mean to be passive? That means that it requires no energy input, and usually that energy source is the form of ATP, right? No ATP, or we'll put energy here because ATP is not the only source of energy, right? Requires no energy. And diffusion follows what we refer to as Fick's law of diffusion, right? Fick's law of diffusion. And if you haven't seen Fick's law of diffusion, there is a formula for it. A equals D star times dc over dx, right? And so what does this really mean is that the rate of diffusion is dependent upon three things, okay? So it is dependent on distance, it's dependent on temperature, it's dependent on the molecular weight, okay? And it is dependent on the concentration gradient, or the energy gradient, if you will. Okay? And when we look at diffusion, this process can either be what we refer to as simple, or it can be facilitated. Okay? So your simple diffusion it's simple. It is movement. I'm going to abbreviate movement here. Directly across the membrane. So these are your non-polar or lipid-soluble stuff. Okay? Very few things can do that, right? Facilitated is where it is carrier mediated. Everybody remember the carrier proteins from the last video, right? Carrier mediated, where you have the membrane, like so, and there will be a protein where it says like this, right? And then that thing can move through that channel, right? So it is carrier mediated or channel mediated, right? So we can use carriers or we can use channels, just like we talked about in the last video. Now, even with our simple and facilitated, right, whether it's directly across the membrane or uses some sort of channel or carrier, we are still talking about diffusion. That's movement from an area of high to low energy, right? High to low energy. Okay, but what if we need to move things against that energy gradient? What do we need to move things from low energy to a high energy? Well, if we're going to do that, that is going to require some form of ATP, right? Or some sort of energy input. So let's talk about what we refer to as active transport. Okay? Active transport, because we actively have to do something, or a body has to actively do something in order to move something across the membrane. So if we look, let's draw our membrane here. These are going to require some sort of protein involvement. Okay, and so let's just make a carrier protein here where we'll do something where it's like closed here. And then I'm going to draw this one like this. There we go. Okay. Where something will bind to it, like so, and then it will sort of reverse in nature, like that, once that thing binds to it, and open it up on the other side. But in order to go from this conformation to this conformation, it's going to require the hydrolysis of ATP into ADP in inorganic phosphate. Okay, so with that, right, this is the direct use of ATP. So this is what we refer to as primary active transport. Primary active transport. Another type of transport that uses energy is known as secondary active transport, and it's a little bit more complicated than primary active transport because it doesn't use the energy directly. So it does use energy, usually in the form of ATP, but it doesn't use it directly. And so in order to look at this, we're going to take a look at the sodium glucose symport channel. So I'm going to draw a very simplistic variation of this sodium glucose channel. And so this sodium glucose co-transport channel can take sodium and it can move it into the cell. And it can take glucose, which is a larger molecule, and it can move it into the cell. Now, this symport channel, when it moves sodium, it does so in a passive way because sodium is moving with its energy gradient. Okay, so this is sort of the movement of sodium is a facilitated diffusion. But what's happening is as this sodium is moving into the cell, it is freeing up energy. It's taking that potential energy and freeing it up, and we're harvesting that energy to move the glucose into the cell. Okay, so the energy for moving glucose into the cell comes from the free energy that's being freed up from the movement of sodium. But in order for this to happen, there has to be a mechanism that keeps the energy gradient for sodium high. So over here, there will be sodium that is pumped out of the cell, and this pumping of this sodium out of the cell requires the input and hydrolysis of ATP into ADP and inorganic phosphate, right? And so this here, this ATP that we're hydrolysizing into ADP and inorganic phosphate, that's the direct energy input to move and establish this energy gradient of sodium that we can use over here to move that glucose in, right? So in primary active transport, like we see over here, your primary active transport, that ATP is used directly in the movement of a given molecule. Over here, the ATP is used to move one molecule, which then assists in moving another molecule. So that is your secondary active transport, right? And these are all different ways that things can move in and out of the cell. So just because I tend to draw things moving into the cell or influxing into the cell doesn't mean that it can't happen in the reverse way as well, right? Things can move from the intracellular to extracellular compartment using these same mechanisms. And these mechanisms are found throughout the entire body. And in fact, that you can find all of these different movement processes in one region of the body. It's the proximal convoluted tubule of the kidney. And we'll talk about that when we get there. But all of these different methods are used throughout the body. So it's important to understand these methods so that when we get to the different organs and we learn about how things move in and out of cells for individual tissues or organs, we know these are the methods that we're talking about. So with that, I will see you next time.